All right, and I'm also, let's see, can't get to the chat. I'm gonna drop a link to the paper that I am talking about in the chat, because I'm not gonna spend a lot of time talking about the methods, the bioinformatics kind of aspect of this paper, because I, in full disclosure, didn't do it. And I don't understand the details probably well enough to satisfy some of your questions. So please definitely take a look at the methods section. And what I'm going to focus on mostly is kind of the biological insight aspect of this study, led by a PhD student in Andy Allen's lab at the time, Bethany Cullity, and a lot of the co-authors here were also at the J. Craig Venter Institute. But this was an experiment that had been conducted, I think, maybe even when, well, Andy was a postdoc. So it just goes to show that the samples that you can keep in your freezer for a very, very long time, waiting for innovations in sequencing to happen, can still be very valuable and yield to nice publications. So this is the paper. And again, I dropped the link in the chat. But the kind of overarching question, and what I thought would be useful to pull out of this particular study as like a little bit of a thought-provoking node kind of here in this conversation is, when it comes generally philosophically to like omics data that comes from the ocean, we're getting more and more high throughput and more precision when it comes to assigning certain reads to individual taxa. But what is the granularity? What kind of level of information is necessary to address different types of questions about how these communities are structured and functioning in the complex ocean environment? So you might approach a question with a need for a lot of granularity. If your question is, what is the population diversity in blooming taxa, for example, you'll want to assign the reads down to maybe a species level or a strain level, versus like a question like, is the whole community resource limited, which may require a different level of granularity that can still come from sequence data. So I'll start out there and then introduce the experiment that was done. I wasn't involved in these experiments, but I've thought a lot about them. And they are 200-liter barrel experiments using seawater from the Monterey Bay, so natural communities. And the experimental design was such that they were meant to simulate blooms driven by upwelling conditions. So in this nitrogen depletion experiment, 199 liters of sub-nutricline water was mixed with 1 liter of surface water, and this was done in biological duplicate. And that simulated a phytoplankton bloom that got over 40 micrograms per liter of chlorophyll A, and kind of ended up crashing several days into the experiment. And the nutrient data suggests that this was because they ran out of nitrogen. That was the reason that bloom crashed. It's a slightly different experimental design with the same community or a similar community using iron depletion. So in this barrel experiment, 200 liters of surface water were used and nutrients were added to it, versus a treatment where an iron chelator was added. So this is a simulation of a phytoplankton bloom by the addition of not subnutricline water, but exogenous nutrients except for iron, and then with the addition of desferoxamine B to chelate the available iron. And these were sampled at 2 time points. So initial MB5, MB7, and then the final MB8 and MB6. MB8 was the iron deplete, and this would be kind of the control. And then the nitrogen had two conditions here. And you can see the chlorophyll, Fe over Fm, nitrite, and nitrate. I don't think we had silica measurements for this. Here is what happened. These are microscopy assessments of the phytoplankton community composition in response to those blooms. The Utermol method was measured to differentiate the types of things that could settle. The big things, centric diatoms and pinnate diatoms, were the things that bloomed in the nitrogen experiment, and the same in the iron experiment, but to a lesser extent. There were other taxa represented that we could see with a microscopy, and that's shown here. And then using the lineage probability index from Dark Horse, I think, was the part of the pipeline that allowed us to assign identities to certain reads that were sequenced from the RNA samples collected. You can see that the majority of the reads are being assigned to centric diatoms in the nitrogen experiment and to dinoflagellates. And this is mRNA. So this, we're calling it kind of total activity. And this is another way of viewing that broken down by taxa. And the thing to notice here, and I'm not going to talk about this really at all, but there were a significant number of viruses or viral reads detected in both the N-deplete and iron deplete condition, which is part of what we identified as terminating those blooms in these mesocosms. Okay, so this is another figure from the paper showing the open reading frames and the amount of reads that mapped. And it's basically a way of identifying, and this is mapping to MMETSP taxa, so members of that project that have sequenced representatives. And you can see just from the list to the right what the most dominant species in these experiments were. This is only the nitrogen barrel experiment, shown here on the right for your reference. And we filtered out the kind of identified it as a positive hit to a reference species if it had a 60% identity or not. This is the open reading frame, and I think that is at the nucleotide level. But I can't, that would be in the methods. I can't totally remember. But what you see is that, 60% identity is pretty low when it comes to being able to confidently identify a species, but it still gives you a sense of how many of those reads in the experiment were mapping to something like the Lasiacyra or something like a Pseudonychia. So it's probably not positively identifying them as that strain, but it's at least something similar. And what these histogram kind of traces are showing is the lighter colors are the later time points. So what we're seeing is kind of a purification in the diversity. So a few taxa within whatever's mapping to that reference are the ones that are by the end of the bloom kind of taking over in that condition. And was what we saw for Lasiacyra and Pseudonychia, but not the case for Prorocentrum, which was one of the abundant dinoflagellates that we saw responding in this experiment. And these plots are showing kind of the breakdown and what some of those functions were assigned as. I'll talk more about, I think, a better way of looking at that kind of data later. Here is another 30,000 foot view of what we actually sequenced and identified in these different barrel experiments. So nitrogen deplete, there were a lot more ORFs that were measured and identified. Iron deplete and the whole iron experiment in general, there was less to sequence. And this over here is breaking it down by taxa. Let's see, I'm going to move my Zoom screen because it's covering. Anyway, you can kind of see these bar plots and what different taxa we were assigned to. So this is still kind of like in a gee whiz, look at all this data. We can identify certain things, and this is how we looked at it kind of space. And these contour plots are a little bit difficult to assess kind of without being walked through them. So I'll take a minute to talk about it. But we had 4 kind of conditions that we were comparing. So all of the open reading frames that were identified, whether it was the nitrogen experiment or the iron experiment, were thrown kind of together bioinformatically and then parsed out based on whether they were differentially expressed, upregulated during nitrogen deplete states, nitrogen replete, iron deplete, and iron replete. So again, this doesn't give you any kind of functional information, but you can see like here, the chlorophytes, for example, almost all of the open reading frames are falling in the quadrant of the plot here down in the lower left. And these were open reading frames that were upregulated when both nutrients were abundant. So when there's nitrogen and when there's iron. Contrasted with something like the viruses over here, so you're seeing that contour. Most of those reads were being picked up in iron deplete and nitrogen deplete. So this gives you, broken down by kind of a certain granularity of taxa, a really kind of quick snapshot about how the different groups are responding to these different nutrient stresses. The haptophytes and the pelagophytes here are really similar in the way that they respond, just because you can see visually that there's a lot of overlap between these contours. And the pennate and the centric diatoms are kind of similar too, where you start to see this big bleb over here that you don't really see so much in the haptophytes and pelagophytes. So it suggests that these different taxa have different strategies in terms of how they use their genome at the level of transcriptome. So most of the reads that are detected, as is probably the case in any phytoplankton metatranscriptome, are the light harvesting complex related genes. And these bubble plots are the same, orienting you in the same way. So all of these LHC genes are really disproportionately expressed when nutrients are abundant, which is not surprising. That's kind of what we would expect, given what those genes are doing in the cell. And this plot is just to emphasize that the amount of sequence data that was recovered from this greatly improved the resolution of our understanding of kind of the diversity of these protein families. Okay, so, metatranscriptomes, I haven't personally done a lot of metatranscriptome studies. Most of the work that I have done has been in model organisms like Phaeodactylum and Thalassiocyrus pseudonana, using the tools that the genome enables. We can do high resolution gene expression studies, both at the transcriptome and the proteome level. We've labeled nitrate and watched the way that metabolites get labeled up within the cell, a fluxomics approach, connected that to our understanding of the evolution of the different protein families in phytoplankton. So when I was first getting involved in the metatranscriptome, I kept asking, well, but which gene is it? Because, for example, Phaeodactylum has 7 nitrate transporters, and they are expressed all very differently in response to changing nitrogen conditions. So when you go out into the field and you see the change in expression of one gene, which one is it? Which ortholog is it? How do you compare that to what we know from Phaeodactylum? So the idea would be kind of using Phaeodactylum as a little bit of like a Rosetta Stone to understand and interpret what the patterns that we see in the field. So this is a paper that we published in 2019 that kind of laid out the groundwork for a very detailed understanding of nitrogen metabolism in Phaeodactylum. I'm not going to go into any of the details of it, except to say that I found being able to incorporate this kind of lab constrained knowledge was really useful ultimately towards interpreting the field data. Some of the other work that has been done in Andy's lab that is useful for this is also in Phaeodactylum. And what this is showing is wild type Phaeodactylum, and then Phaeodactylum with the nitrate reductase gene knocked out. So in this condition, they're able to transport nitrate, but they can't actually assimilate it. So they get these big swollen vacuoles and they look like bowling pins. And paradoxically, they're experiencing a state of nitrate repleteness because they can sense that there's plenty of nitrate around, and the cell, if there are mechanisms to sense that they have nitrate, they know that they're not starving. But physiologically, the cells are actually starving for nitrogen and showing kind of hallmarks of that physiological condition. And we've looked, kind of in explicit detail at the expression on high time course resolution. So we have a pretty good understanding now of what pathways are co-regulated and kind of what conditions they are uniquely responsive to. And again, I'm not going to go into too much detail about this, but this is a time course where cells were exposed to no nitrogen 1 hour after a nitrate spike. We know that at 18 and 42 hours, there's no nitrate in the medium, but the cells themselves are replete for nitrogen. And then after the 42-hour time point, they become starved for nitrogen again. So you can see that these genes, this is the nitrate assimilation regulon, is highly upregulated when nitrate is present, but it's also highly upregulated when nitrogen is deplete. So if you were trying to interpret the expression of something like a nitrate transporter, that complicates the story because they are, those genes are upregulated when nitrate is around, and they're all at the transcript level, and they're also upregulated when there's no nitrogen around. So, if you measure high nitrate transporter transcripts in the field, it doesn't really help understand what the nitrogen status of that overall community might be. So to kind of incorporate what we know about phaeodactylum into this data set, we did an ortho-MCL analysis where we took all the phaeodactylum protein models. And again, I can't remember if this was in nucleotide space or amino acid space. I think ortho-MCL requires amino acid space. So I think that's how it was done. And we had about 200,000 ORFs from the nitrogen and the iron experiment that were assembled into 11,000, roughly 12,000 clusters with the phaeodactylum models in there. So we could go in then to those clusters and I could be like, okay, this is the one that we know is responsive to nitrate and nitrogen. And these plots are really similar to the design of those other contour plots, except that the circles now are showing ortho-MCL clusters. The big ones are the ones that had more ORFs assigned to them. And unsurprisingly, the light harvesting complex genes here are shown in the quadrant. This is only for diatoms, by the way, that are shown in this quadrant are upregulated when the nutrients are abundant. But there are a few of the photoprotective LHCs, like the LHCX1 gene, that do pop out as being disproportionately upregulated when iron is not available, and also a little bit when nitrogen is not available. So this is the average. I don't know if this is intuitive or not, but the value that's being plotted here is the average log2 fold change for all of the significant open rating frames that were assigned to that ortho-MCL cluster. There's a couple of interesting things to note, like not a lot of genes are upregulated when they are deprived for nitrogen, but it was kind of a good sanity test to see that the nitrate transporter gene, and the one that we know that phaeodactylum has that responds this way, was upregulated when nitrogen was deplete, along with a whole bunch of other ribosome transcripts. And counterintuitively, if you went out into the field, if I did, and measured and saw a whole bunch of ribosome protein transcripts being upregulated, I might interpret that as, all right, they're upregulating their ability to make protein. But more likely, this is that condition where they are upregulating the transcript when they don't have it, as a way of kind of front loading. I think Rob Lampe's paper articulated that concept about kind of having these genes expressed at the transcript level, ready to go when the conditions are favorable again. And this is something that we kind of see uniquely for diatoms, not so much in the other taxa. So this is breaking down metabolism even further. I'm not going to get into detail so much about that. But some of the genes with very specific function that we know to be important in the nitrogen physiology from the controlled lab studies on phaeodactylum also were popping out in this analysis. But I want to point out that this is the nitrate data from this exact experiment. And the nitrate, and this is in micromolar. So it goes from about 20 micromolar at the initial time point down to, I think, 12 or so by the time that the experiment was being sampled. So these communities are not nitrate starved, and they're not even really nitrate limited. We know that nitrate availability or nitrogen availability ends up terminating the bloom in conjunction with the viruses. But these transcripts are very, very sensitive to lowering levels of nitrogen availability. So what we're seeing is that the gene expression patterns are telling us more about how the community is preparing to respond what they anticipate the next condition might be, which is total nitrogen drawdown and lack of nitrogen resource. Okay, so these two genes are, the nitrate transporter is one that we have a really good handle on how it behaves in culture. And a gene that's a couple steps downstream of it, which is called glutamine synthetase 2. This is the isoform that is plastid localized and involved in assimilation of nitrate, has a really similar pattern of expression in culture. So this is a time course experiment where we spike in nitrate. Both of these genes increase. This is, I think, FPKM or RPKM. And then there's this period of time where they're both repressed while they have internal reserves of nitrate, or they're kind of physiologically not depleted yet. And then the nitrate transporter increases as nitrogen is no longer available and they become starved. But GS2 doesn't do that. So GS2 has the nitrate responsive trait characteristic, but does not show the dramatic upregulation at low nitrogen that NRT2 does. And if you plot that as a ratio, you can see that the ratios that are above 2 are really kind of exquisitely tuned to pick out a nitrogen replete or nitrogen deplete cellular state. And this is kind of appealing from a methods development point of view, because now we're able to rely on just 2 genes that don't need to be quantitatively measured. Like you could go totally rogue and not even measure how much volume you're filtering, as long as you get the from the same sample a measure of one transcript abundance relative to the other. You can make that ratio and get this kind of snapshot about what the whole community is experiencing. So this is a slightly different way of looking at it, broken down by diatoms and other taxa. So the nitrate transporter is here in the blue box and whisker, and the GS2, the isoform that we know to be nitrate responsive, is here. So what you're looking at is above the line and below the line here in the gray and the blue. That pattern holds for other taxa as well, chlorophytes and haptophytes. Dinoflagellates, not so much, just because there was less dynamic expression of these genes in response to our experimental manipulation. But the ratio, this kind of proxy for nitrogen status, may hold outside of other taxa except diatoms. If you break it down to an individual species level, you can see that we've included some of the other studies here. We've got N-replete and N-deplete measures of this ratio, and it tends to follow what we expect it to follow in a variety of different conditions and a variety of different taxa. So, and we wrote, this is an excerpt of the little paragraph that we put in this paper. And the reason that I'm talking about this even is, what is the value of these molecular proxies in a sequence everything world? And I think over the last maybe 10 or 15 years, the innovations in sequencing, our ability to get longer and deeper and kind of more phylogenetically resolved, has done such phenomenal things for the field. But there may be now a moment to kind of transition back to a little bit more of a simple metric, depending on the questions at hand. And we still, in biological oceanography, have a need for assays that are quick, that are simple, that are cheap, that can be done at sea, that can be done in a way that can increase our spatial and temporal resolution without having to sequence an entire, metatranscriptome every time. So this proxy, NRT2 to GS2, there's some work being done in my lab right now to kind of flesh that out as a way of being able to go out and kind of rapidly sample and be like, is this community nitrogen limited or not? And to inform like adaptive sampling that might be happening at sea. You could be doing this with like a qPCR machine and a really rapid one-step extraction on board. And, you know, some of the work that I am getting more involved in is using ocean iron fertilization for carbon dioxide removal. And is that a good idea or not? I'm not going to get into that because I don't really want to have that conversation. But in the next generation of experiments that are going to be done, or would need to be done to ask some of the outstanding questions, we're going to need tools like this that can inform sampling that can be done adaptively and in response to tracking a patch, for example, or observing the evolution of an experimentally initiated bloom in response to an artificial iron addition. So that's kind of just my two cents about it. I love sequencing everything, but it doesn't need to be done all the time. All right, so hopefully I've stayed more or less within my time, and I will leave it to the moderators to manage questions if we've got them at this time.
Yes, thank you very much, Sarah. We do have time for one or two questions, and then also hopefully we'll have some time at the end of the second presentation for further questions for both our speakers. Is there anybody has a question right now for Sarah? I believe you should be able to unmute if you would like to ask a question, or feel free to put in the chat, and I can read it to Sarah. Okay, we do have a question from Sasha. You can go ahead, Sasha. I believe you can unmute and ask.
Hi, Sasha.
Hey, hello. Hey, yeah, thanks for your talk. It was nice to go through this data again. Yeah, I like the idea of the ratio. I've been looking at ratios instead of like absolute abundances as well in different in different field. But what you said in the beginning about at which taxonomic level it's like appropriate to look at things. And when you showed these ratio things, it reminded me of a work I did that I had to scrap because I was looking at metatranscriptome data at species level, and we updated at one point our reference database and updated all our data, and then sometimes there were some shifts at the species levels of like where genes were annotated, and whole pathways would just fall away from my species because when they were first previously annotated here, they were annotated at different species, within the same genus, for example, but a different species. And all of a sudden, I would have species that would have like partial metabolic pathways that were not complete. So I was thinking, when you do these kind of ratios, even of looking at just two genes, I think the difficulty would be to make sure that you are looking at the same, or that you choose the right taxonomic granularity and that you compare the right things together. Because you could, in my case, when we switched the reference database and our taxonomic annotations, the storyline just totally changed. And in the end, I stopped that work and I just took a big step back and like, okay, now I'm looking at a much higher level until we have more, better ways to get those taxonomies really nailed. So yeah, do you have any thoughts on that?
Yeah, I think that's a great point. And yeah, you never want to kind of overinterpret beyond what the technique itself can actually handle. But I think, the nice things about those two particular genes is that they are really highly expressed, and every diatom has them in at least the GS2, they have it in mostly like a single copy. So it's a pretty robust of all of the things that you could be looking for. But my kind of way of coping with that problem is to just like blur my eyes and zoom back and be like, all right, let's just measure it for all the diatoms, because the ones then that will give you kind of a sense of how the diatom community is responding. And we have some key PCR primers designed for conserved regions within the nitrate transporter and the GS2 sequences that could be universal enough for diatoms. So you're losing the ability to say something about like which strains or species within the diatom group are experiencing nitrogen limitation, but you're gaining the power to say something about the whole community, which is more than I think we can do with any other technique.
Yeah, yeah. Actually, now that you're talking, I'm thinking if you are focusing just on 2 genes, you could probably get a lot of, really good phylogenetic trees of those 2 genes, and actually could pinpoint pretty well those taxonomies, because if you would place that on your very extensive trees or something like that. So you may even go actually really in the other direction because you're only looking at 2 genes and not at metatranscriptomes.
Yeah,
If
that's
you
a
want,
great
yeah.
point. Somebody give me funding to do it.
Yay!
I'll keep trying.
Thanks.
Thank you.
Awesome. Thank you, Sasha and Sarah. Well, speaking of reference libraries, let's move on to our second talk. And I think that this will probably spur other questions that are similar. Maybe we'll have a little discussion later. Because that's a great question, Sasha. So now we're going to hear from Mora Groussman. They're a bioinformatics professional and former postdoc scholar at the University of Washington. Got their PhD in oceanography, master's, BS, all from the University of Washington. So we have a strong West Coast presence today, which is great. And they're going to talk about their MarFERReT eukaryotic sequence library. Go ahead.
Hi, everyone. Thanks for having me today. Let me just get my screen up and shared here. Okay, does this look good? Hi, thanks for the introduction. I'm Mora Groussman. Whoops. I'm a former postdoc at the Armbrust Lab. And again, you heard about the PhD at the University of Washington before that. And I'll be talking like Sarah said, about reference libraries, and in particular, the MarFERReT eukaryotic sequence library, and a little bit about what spurred it, what went into it, and how you can use it and customize it for kind of a flexible but reproducible reference library framework. Okay, so if you want to read the paper, this has been published in Scientific Data, and so you can find that there. This came off the press in December of last year. And it's also tightly linked to a GitHub repository where we have the code base and documentation, and Zenodo data repository where we have our big files like the protein FASTA files themselves, annotation files, and other things like that. And actually, I'll do the same thing that Sarah did here. If I can manage it over Zoom and drop some links in here. All 3 of these. Here we go. All right, so there's some links if you want to take a look at those at any point. I'll go back to my screen here. Okay, so they all link to each other too, so if you find one of them, you can probably find the others. Just to go over what I'm going to talk about today, I'll be talking a bit about the motivation and goals behind this project, and then how we went about collecting and validating all of these different entries, and then talk about the contents in the latest MarFERReT build. And then we'll talk about how we can use it, either just as it is, or replicating the build process, or using different custom approaches, and how to expand, modify, or integrate it with different reference libraries. And then how it can expand in the future by contributing and enhancing open source code base on GitHub and Zenodo. Okay, so what's some of the rationale behind doing this? I've been doing metatranscriptome annotation for a while, and there's a lot of great resources out there. It kind of got started when the MMUTSB data just started to come off the sequencers and become available, and that was really exciting. But lots happened since then, and it's been a little tricky to keep up sometimes. So there's some taxonomic reasons that would encourage us to do this. Some of us have recognized that some lineages are underrepresented in the current databases, especially those tailored to marine microbial eukaryotes. There's some gaps that we've noticed and some underrepresentation in, say, open ocean lineages. There's also a good proportion of metatranscriptome data that doesn't really get any significant or confident taxonomic annotation. And by filling in those gaps, we can get at that kind of unknown fraction of biological dark matter. And sometimes in the way that other libraries are prepared, they don't often maintain all the levels of granularity, sometimes dropping off the strain level or species level, which again, whether we want to work at that level really depends. But for something like Pelagococcus, which is cosmopolitan at the species level, it might really depend on what strain we're looking at. This project is also an attempt to address some issues that me and my team have noticed in updating and reproducing sequence knowledge. How do we integrate all the new knowledge that comes out year after year into these static libraries? Some of the compilations that try to aggregate different resources don't always have a DOI or a publication available, or documented code is not available. And we all know that sometimes we have, say, a handful of our favorite transcriptomes and genomes that we want to add in there, and then what happens to it? So a lot of us have seen these custom reference sequence libraries that is really this mismatch, but that's not reproducible. So this is a MarFERReT was the project that came out of one of my PhD dissertation chapters to try and address this. The primary goals are to, well, focus on marine microbial eukaryotes comprehensively because they often get left out of, say, prokaryotic focused libraries as well. Update it with as much new reference sequence material as we can grab at the time, although it could also use a refresh already. And then also curate the metadata, the NCBI tax IDs, the names, and then validate and quality control the entries themselves to pull out anything that might be affecting our results in a negative manner. And then put it all in a reproducible code base with accessible data products and a simple and straightforward framework for incremental updates in the future. All right, so let's give a walkthrough of what's in here. So I collected 902 candidate entries, and these entries can be transcriptomes, genomes, single amplified genomes, and nucleotide or protein format. And then the nucleotide sequences are translated to course everything to a protein sequence format. And it's gathered from a wide range of different things. Most of you are familiar with the MMETSP and the reassemblies. But there's a lot of other things out there that have been sequenced and made available since then. Roscoff culture collection from the MetDB, 41 more entries from there, a handful of things from NCBI GenBank, 116 from JGI Phytoplankton, and a number from smaller novel sequencing efforts that might not be included in some of these larger repositories, like some oligotrophic open ocean isolates from some of the North Pacific study regions that we go to, Antarctic diatom isolates, uncultured single cell genomes, and a handful of others. So we're not only trying to get some of the big stuff, but also a lot of these kind of smaller sequencing projects that really would make a valuable contribution to the reference sequences that we have. And then once we all have them protein space, I'm skipping out a lot of the technical details. This is all available online just to try to get at the high level functions here. As we annotate them with Hammer and PFAM, just to get a sense of how well do they encode proteins as we might expect them. So there's also a lot of manual curation in here too, in updating all the NCBI tax IDs to make sure they're up to date, the names are up to date. And you can see this here for the NCBI lineage. And we know that's kind of flawed, and some of us like to use it. A lot of tools use the NCBI taxonomy, and a lot of the software that I use goes that well too. We've also linked it to PR2, which is the Protus Etinus ribosomal catalog. And it's important to emphasize matchup 1-to-1, but this is just kind of an accessory match for the best match that we could find for each one of these. And this is all in the entry metadata, and helps us bring a lot of that metadata back up to state. Now, these are kind of boring spreadsheets, but one of the core documents, we have this metadata file where we have all 902 entries, every one of these files. And there's a lot in there, but if you start to work with this, there's just a handful of critical fields that you really need for the pipeline. An internal entry ID, the name, the NCBI tax ID, and the file name that it points to. And of course, whether it's a nucleotide or amino acid, that influences how it's processed. This accepted field is important because our quality control measures that we put in place, we still keep them in the metadata, but we may not choose to include them into the final build if they don't meet our quality control criteria. So that way we can keep track of what we've looked at, but not necessarily decided to incorporate it into our reference library. There's more fields that you don't need to run it, but you probably do need to publish it, such as the link to the source, reference publication, and the data citation DOI. And these are all good to just kind of have and collect if you start, say, adding to this kind of thing yourself. There's also a bunch of non-essential yet helpful fields that just capture a number of different data and parameters, kind of like what data type, data source, publication year. The pipeline can run without these, but these are nice to have for looking at things. And also the best match is the PR2 database. Again, it's not one-to-one. We try to find the best rank match that we could to try to link amplicon studies and metatranscriptome studies. And also some people do prefer the different taxonomy structures in PR2 over that of NCBI. Okay, so I'll gloss over the quality control process. This is all written up in depth. But we flagged candidates for entries if they had less than 1,200 total sequences or less than 500 unique PFAM domains. So if they didn't really show like they had much protein coding potential in the gene models of the transcriptomes. We also incorporated kind of a technique to pull out the ribosomal proteins from all these entries and then do a lowest common ancestor analysis to see what it looks like they are from that perspective. And some of these have been flagged as possibly putatively contaminated from additional studies, also turned up with this method as well. But this is something that kind of helps us flag things that don't look quite right. So there were 102 entries there that were flagged, which is a chunk of, say, 12 to 15%. But if these ones aren't looking really good, then we don't want them in our reference data. So it leads to the final selection of about 800 entries. Over here on the right looks like a phylogram or cladogram, but it's not. This is actually just a hierarchical clustering of the presence and absence of different prefam families, which kind of recapitulates phylogeny. This is just kind of a visual diagnostic, but what's marked here are some of the different clades where we've excluded entries. The blue ones here from our own ribosomal protein analysis, and they tend to line up with these red and yellow dots from a Van Bleerberg paper and a Lazek and Nyskos paper that analyzed MMETP samples and some ciliates from the NMETSP and assess contamination issues or putative issues. So I feel like these, they could use some more analysis, but for the sake of safety, why not leave things like this out, or at least apply some level of stringency to what we include in the final build. And this is all available also in the web resources in a curation CSV document here for the latest version that can tell you all the parameters that went into that and what makes them look not so good. It's written up, again, more in depth on the online documentation. So what do we have in the end? In the final MarFERReT build, if we take with these 800 entries of genomes and transcriptomes and SAGs and SATs, here in this figure on the left, this is a cladogram of the NCBI hierarchy for these 453 NCBI taxides at the species of strain level, representing about 28 million proteins that are clustered within each taxa, not as a whole. And you can see the branches are colored by the different lineages, some of the major phytoplankton lineages that we all know and love. And the outer rings here are just kind of some metadata indicators. The green ring is kind of when these data for these different sequences came out. You can see it covers things from 2002 all the way up to 2021. So there's a lot of exciting stuff that's come out then too. The number of sequences for all these taxa, and also the data types, transcriptomes, genomes, some single amplified genomes and transcriptomes. So the mixed one is a taxonomy that might have a genome and a transcriptome, for example. And the outer ring is the data source, including some of our larger ones like MMATSP, JGI, the Roscoe's Culture Collection, NCBI, and others. All right, so if you want to use this, what do? There's a couple different ways to go about it. And a lot of this depends on what are your research needs and what are the tools that we use. In our lab, we use Diamond a lot for taxonomic annotation and analysis. It's pretty fast and go through a lot of proteins. And there were some MarFERReT data products that we put up on the Zenodo repository that are ready to plug right into that. The protein files and also some taxa mapping files. So on the GitHub documentation, we have a lot of stuff laid out there, including how to use it directly. You can just download some files from the Zenodo repo. And then if you have something like Diamond installed, you can use it to annotate your non-environmental sequences or what have you. This is a diagram I pulled showing one of these annotation pipeline workflows. And over here, you can see the MarFERReT proteins and metadata that we have in the data portal, along with some NCBI taxonomy files that you can get from NCBI taxonomy and GenBank. And these are built with Diamond into a database that we run to annotate kind of like fast blast. They can also, but we tend to use it with lowest common ancestor analysis. And this can be combined with PFAM or COFAM or any other functional analysis to do any number of fun statistics and results that you have in mind. But in addition to just downloading it as the final product, we've also included a complete pipeline that you can clone from the repository on GitHub. It's implemented mostly in Python and Bash with just a handful of R scripts. And it also shows you, tells you exactly where we got the data from, so you can collect it yourself. And we've built some containerized scripts with Singularity or Docker containers that can, with just a single line of code, start to build and replicate these to get the final data product again. And this is also where customization comes in hand if you want to tweak the contents of the repository or the library. So we have it as is, and we've shown what we've done, but a lot of us have different things that we want to do. Maybe you disagree with the quality control criteria and you want to make it more stringent or less stringent. This is something you can kind of tweak. This would make it a different version and you would have to name it differently, but this gives you kind of an easy option for fine-tuning what you want to look at. And a lot of this, you just have to change. If you don't want to change the entries that are in there, you can tweak some things in that core metadata CSV that will then be parsed and run in the machine scripts. But a lot of us want to add more things too. Maybe you have 5 or a dozen new isolates or culture isolates, and you've got their fresh transcriptomes and you want to add them to this library. And this can be done through the same framework. If you have the gene model files in FASTA format, add them as new lines and new additions to the metadata file, and then you can run them. Or go through the GitHub framework to try to collaborate and get them in public inclusion for a public release if you want to add them to kind of a shared or common framework as well. But in addition to adding a line by line too, it was also kind of constructed with the sense of merging it with other libraries that might have other specific domain focuses, such as those prokaryotic libraries, or fungal libraries, marine fungus, or anything else that might complement this. And a lot of our data could really use this. So I have one example here that we've used in a project. where we have MarFERReT, which specializes just in marine microbial eukaryotes, and we combined it with MarMicroDB, which is mostly prokaryotic focused, but both of them converge on NCBI taxonomy. They can be merged and then used with Diamond to annotate sequences both for eukaryotic and prokaryotic annotations. So how do we continue using this? There's a lot that we can do. I've got a lot of ideas. There is a wish list in the works for new candidate entries that I've worked with, and Sasha also has her hands on. So if you get a chance to talk to her, or if you have any ideas about what can go on there, I'm sure we can think of a way to pool this list and see if we have some shared ideas or if things missing. And a lot of the reason that I put this up on GitHub is so that it's not dependent on a specific repository that relies on a funding cycle or a specific staffing cycle. Now it's out there and it's open source software, and there's a couple of different mechanisms that people are going to use to either contribute to it communally or take what works and use it for themselves, such as issues and pull requests from the GitHub repository, or repository forks. If you want to make some serious changes, fork all the code and then do whatever you want to it. But I think this makes it easier to keep track of what we do, what we add, and also what we publish. And hopefully others will find it the same. Okay. And okay, so that is yeah, that's at the end of the slides. So thank you. I want to thank my collaborators on this and co-authors, Sasha Cassell, who's here, Stephen Boskowski, who wrote some of the great containerization scripts, Ginger Armbrust and the rest of the Armbrust Lab, and all of our scope collaborators, and the School of Oceanography and Simons Foundation. And also throw a little mention out for myself. I am looking for work. So if you have any interesting projects in mind, please reach out more at jovian.bio. Okay.
Thank you, Mora. So I'm not sure, can you see the chat? There is a question from Ariana, and I just, rather than me just reading it, it's probably, if you wanted to just answer that question.
Yeah, I'm just getting a chance to look at them now.
Okay.
That's a great question, and that's is it possible in the future to clean contaminated resources to retain content for more of the references? And this, absolutely. And I think if I had to go back and make one change or improvement that I couldn't quite get in, this is something that I would tackle. To assess cross-contamination for the paper, we looked at these ribosomal proteins, 63 of them. But, we really have the compute power these days. It's accessible to do something like that for all of the genes and see at the very base level, does it look like this is falling in the right phyla? And I think some of these kind of problematic looking transcriptomes or genomes could be cleaned up in this way. And I think a couple things to take with nuance too is what kind of contamination is it? Is it something that's really kind of icky, like a fungus growing in the laboratory and cultures, or is it actually something biologically cool and interesting? And I think a lot of these contaminations that is really what they are, because we have these chimeric entities that are a fusion of, say, a dinoflagellate and a diatom, and they're going to look contaminated because they are chimeras. So I think it's important, if we were to go and improve this, to one, see if we can assess, yeah, if it doesn't belong in there at all, for example, like a ciliate in some culture that is their prey, then check it out. Or some way to partition these two. For example, if you have the diatom dinoflagellate symbiosis, for example, why not make that 2 entries and kind of partition the dinoflagellate sequences from the diatom sequences from the things that don't look like they really belong anywhere. And I think if we were able to do that, that would really help the interpretation of our results. And I think it's absolutely plausible. To answer that again, yes, I think that'd be a good idea moving forward.
Are there other
some of them note that
questions?
it's because of the prey, and it's the same thing there too. I think this, I think it's really tractable to go and just separate sequence by sequence. Does it look like prey or, and I think that has been done, but but applying that approach again and again to things that come in a standardized way would help us kind of make sure that this is done beyond MMEDSP.
We have a hand, Sarah.
Yeah, thank you for your talk, Mora. I appreciate the attention paid to building a reference database. I had a question. I noticed I was using MarFERReT actually not too long ago, and I also noticed that you have the PR2 reference ID to it, and that's kind of like the gold standard for amplicon sequencing. And I think a lot about how those two types of data can be, taken, if they're taken from the same sample, like what can we do with that information? And I just kind of want to ask generally, what did you find when you tried to like correlate the transcriptome stuff that you found from various databases to the PR2 database? Sorry.
Yeah, that's a good question. And I think a lot of this gets into the nitty-gritty of how the NCBI taxonomic architecture kind of aligns with PR2, how they clash and how they collide. So PR2 is the gold standard, and if you look at the PR2 metadata files, they have what essentially goes to an NCBI tax ID. But the way that they organize their taxonomy lineages, their levels, only goes down to the species level, which is good for plenty, but not others. So I don't have the numbers quite at hand, but for a lot of these, I was able to match them one-to-one. The culture to its representative in PR2. And again, some of them are missing. There might be some genome that doesn't, or something that doesn't have the exact representative in the PR2 database or vice versa, but most of them did match. But it was a little fuzzy to try to get them matches some of the ones that had different strain IDs. So for this bulk approach, I kind of did this match as well as I could. And then I employed like a fuzzy string matching thing to try to see if there's like CSCMP 1338, match keywords to a deeper level. And in others, you could only find a match to the genus level. Like there might be something that has a species level representation in MarFERReT, but only the closest match is at the genus level in PR2. These are just a handful. Most of them, however, those are kind of the edge cases. By and large, they do fit pretty well. I just took a bit of a manual search to say, okay, what is the best match we have? And that's in the file too, is what level we were to match it at. Is it matching at the species level, the strain level, or did we only find something at the genus? But overall, I think, yeah, it could help kind of tie those two worlds closer just a bit more.
If I can sort of follow up on that, we went through a process of trying to find functional genes that provided a similar level of phylogeny as, like, for example, 18S. And we had a hard time trying to do that, because I mean we kind of figured, well, maybe what we can do is if there are particular genes that we know that are functional that come out within the the metatranscriptome, we can use those, and we don't have to sort of also do DNA analysis of 18S or whatnot. Do you have any thoughts on whether that actually could be an applicable approach in relation to how then we can use the metatranscriptome for more detailed taxonomic resolution?
Yeah, and I think a lot of this, might have to depend on what's coming to mind is the different phylogenetic studies that I've used concatenated genes, things that are fairly ubiquitous, like the ribosomal proteins. But again, I don't know how much specificity there is enough for those, but if they're concatenated or combined, although that can get a bit tricky for eukaryotes if they have multiple copies of things. I think those genes are out there. I think I might take a bit of digging to see which ones are really good for that. But it might also be that there's a lot of them that can be used in bulk. A concatenated assortment of, say, 60 ribosomal proteins or these other core metabolic genes. And that was a part of MarFERReT. Is one of the things that we talk about in the paper are these core transcribed genes, where we took the subset of transcriptomes and for, say, diatoms or haptophytes, what's the core set of, say, PFAM domains? And you could do this for CoFAM too, that we see in, say, 95% or more of diatom taxides. And that kind of thing might kind of give us a handle on the ubiquitous genes that, if we dig into them more, have some sort of enough variation or at the right level to really help us pull these things apart.
Mm-hmm. Okay, so if there are any further questions for either Mora or Sarah, feel free to raise your hand or enter it in the chat. Yeah, you can go ahead, Sasha.
Yeah, I guess just to continue a bit this latest discussion, I can imagine if you take like the CTGs, like the core transcribed genes, out of your metatranscriptome data, you could do phylogeny. You can make a pipeline, just make phylogenetic trees of like what kind of what Mora was saying, like of, I don't know, 50 proteins, for example, and get your phylogeny from there, like just adding more instead of like having one very precise 18S sequence, getting your assurance by just having a lot of different like proteins or genes even.
Yeah, and I wonder, you know, for the common example that comes to mind for me is like how, you know, for prokaryotes, they're using a lot of REC-A, which is, you know, another sort of single copy gene. For these CTGs, do you have to sort of be mindful of whether they actually are single copy or multiple copy?
I think, yeah, absolutely. Especially if you do something like concatenation, then if you have multiple copies, you have to find some way to choose, well, what's the right one? This is something that I leveraged for the ribosomal proteins, because there were multiple copies for many of these, but they, when kind of run through an annotation pipeline, would fall out differently. And that was kind of what we got some of the contamination estimates for, either contamination or actual real stuff with predator or prey involved in the culture, or symbioses inside the organism. Yeah, so I think we can
there
have
are
those
genes, right?
apart, but it's also this thing where if they're not single copy, we kind of have to choose, well, which one's real if there's multiple copies. I know that the eDNA community really looks at cytochrome oxidase as a really popular one. But that might be, I'm not sure if that's really more animal specific or how well it applies to the bugs that we look at. I see Eric's comment too about CoFAM, and that's great. Yeah, I think something like that could, if we get in touch with myself or Sasha, get added to some iteration of what's going on. Maybe this is something that we could talk about when people get together is, what do people want to add or see? What's missing from this?
Okay, so there's a comment here, it's just from Eric. In our hands, we find DNA-dependent RNA polymerase genes a good candidate, as they cover both pros and euks and are quite large in phylogenetic signal. Okay, Ariana, if you want to go ahead and unmute, you can ask your question.
Yeah, I was just continuing on this thread, also wondering, since in MarFERReT you have both the taxonomy and the function next to each other, so in addition to these studies that have these genes that for one reason or another might be conserved, commonly transcribed, something of that nature, whether how difficult it is to query the database that exists that you've put together for functional genes that very frequently come up in many resources, whether you're able to look at that in a cohesive way across all these different sources to identify, if not, conserved or important for some reason, what genes you have overlap in presence in each one of your references that's included in MarFERReT.
Yeah, that's a good question. There's a couple different approaches that I think you could take to that. I'm taking a moment to think here. So I think there might be different, when we annotate these things functionally, there's a lot of different tools we use. And with HAM, we're using something like CoFamScan and PFAM, we get these domains, which are they're kind of blocky. It's like looking in the fork drawer, but it could be a salad fork or a steak fork or whatever. PFAMs are a bit chunky, where we might need a little more fine-tunedness in order to find out what they really did. This is something that Shiri Van Graaf did with the flavodoxin. If you look at that, that's interesting. The iron story is that flavodoxin is something that's mostly represented in PFAM space by just one PFAM. And we know, kind of, even though this hasn't propagated into the PFAM space, that it's more complex than that. They do different things. But what Shiri was able to do, and a lot of us have done similar things, Sasha has done things for like this for the light transporters, is use PFAMs to get kind of a bulk chunk of, hey, this is things that look like, say, forks, belong in the fork drawer. And then create alignments or use different tools that may or not be present in MarFERReT to try to get at, well, how can you break these up or categorize or hierarchize them to get at the ones that, how many copies are there? So I think it might be a good handhold to do that. And that's what I did for the ribosomal proteins. Pull them all down with PFAMs, but knowing that there are different copies, and the different copies are different enough to be annotated under different phyla, even under some cases. Most of the entries looked really good, but this did catch a couple of them. And so I think that, yeah, they're clumsy, but they also give us some leverage to kind of take chunks of what things that look like similar, and then do finer scale work from there. Really, there's a lot of interesting approaches that we could take to figure this out further. Did I get your question okay?
Yeah. I mean, I think it's a complex task. I was wondering if there was just infrastructure in the database or things that you've done in the past, and it sounds like you've done a little bit of that, of looking at these different types of functional annotations and then trying to zoom in on where you can and can't draw parallels across the whole database, but just whether there was something that you were thinking about as far as an architecture already within the database to try to tackle the problem.
I'd have to give that some thought to how to do that kind of more technically, but I think, yeah, that's something that could be feasible and mapped out, and kind of gives us a good handhold on teasing things apart, and yeah, kind of improving this this reference, whatever we use as a reference. This is really important, and the more we can kind of fine tune and improve it, and both expand it and also be more stringent about what we include, that'll improve our annotation results. I see another note from Eric about the stop codons. So this is then we've talked about. This is kind of one of the things that is on the perennial to-do list. Again, if I had funding, I would love to deep back on this too, because this is kind of a fun and interesting project. I don't have immediate plans, But this is the kind of thing that I hope would be easy, say if you, because we have it on GitHub in the assembly script, you know, you can find the function or the script that does the translation and the frame selection. And this is where something like this could be then changed or replaced with something that either the community agrees on. Maybe we should do something about the stop codons, or do something about the way that we organize the translations. Or if you just want to go out on yourself too and do it, you know, the code is there. So if it's useful to you, but you don't, but you want to make some tweaks, I think that's what I hope the utility of having this all up in an open framework is, that you could just say fork it or take what works for you. And then if there's some small bit, then yeah, I think, and this is something we thought about, there are some things that could be tweaked for the codons, but I guess, yeah, short answer, no immediate plans.
So while we wait for others potentially to have questions, I do have another question for you, Mora. So I'm in thinking about what you talked about in terms of the different approaches in terms of adding to the reference databases, which I think, you know, has been an ongoing challenge. And I recall the days when I was in Ginger's lab, how everything was about, you know, camera, and camera was going to have this, you know, basically this method of integrating new transcriptomes, and it was, you know, there's a lot of resources behind it. But, you know, as we know, camera doesn't really exist anymore, versus what might be sort of maybe considered more of a community sort of organic or, you know, bottom-up approach of just figuring out ways to share sequences. But I mean, I think, you know, I think probably what would work best perhaps is maybe something in between, because I think there still is this sort of gap in as a community, our ability to know where we can go to deposit new taxonomic or, you know, sort of transcriptome databases. And I was just kind of curious, what are your thoughts, given, you know, the development of MarFERReT and using these sort of public databases, in terms of how, as a community, we can move forward so we can keep adding as people sequence more? And, you know, because again, we're often confined by the reference databases. So do you think that there's a way that we should try that we haven't yet in order to provide this as a community resource?
Yeah, I remember camera too, and it was cool, but it was frustrating, you know, when a whole portal goes down and then it's like, what do? You know, what happens to the metadata that was there and all the different iterations of it? This is part of the hopes of putting up here, is that, well, you know, maybe GitHub won't last forever, but, you know, or Zenodo or NCBI, right? But the bet is that they are more stable repositories. They're not going to be dependent on these ephemeral funding cycles. And I've seen, you know, what happens in the open source world. You know, people put things in the repositories. If people like them, you know, they contribute to the repository, or they change it. They make an issue request. They submit, you know, a pull request if they want to contribute to the original, or they fork it and do something else entirely. And so that's kind of thing where, yeah, I put it out there, but really whether it takes off is a matter of, you know, we can keep improving it. The members that are in the Arborist Lab can put out these iterative releases, but it's on GitHub. You know, this is a community resource by the fact of the architecture it's in.
Mm-hmm.
So I hope putting it out there and having things really laid out on Zenodo too, and also what we've done to try to make things containerized, so you can rebuild it and hopefully get the same thing. But I don't think there's a one-size-fits-all answer too. That was part of my approach, is that we don't have to have the definitive, this is it, you know, but rather some way of thinking, can we keep track of what goes in here and how do we process it? So I think as long as we're finding some ways to both keep track of what we're doing and what's in our reference libraries, you know, no more custom reference libraries. But, you know, we all have stuff we want to add. We have our, you know, 5 new transcriptomes that came out this week. How do we add those in there? So this is my attempt, struggling with your camera and everything else, to be like, well, this is one way, we just kind of keep it in a big spreadsheet. But this, the metadata spreadsheet is kind of human readable. 1,000 entries, we can add more of them. That's one approach to curating our small slice of this microbial eukaryotic world, because we get left out a lot of the prokaryotic focus things or the all eukaryotes. And I think there's a value to having that kind of expert level focus and saying, hey, here's the things that we want to have because we know they're important in our study environments. So we build them in there, and yeah, I think if it's useful, I think it takes off and people use it and adapt it. So hopefully that's what will happen here. Or if someone wants to change something, I really think this is something that people can take ownership over. Make pull requests if you want to, if you have an idea for changing this or that, or again, take it and spin with it.
Sasha, did you have a comment or a question?
I mean, to follow up, like for example, we had a Shiri graph in our lab, isolated a whole bunch of new diatoms on the last cruise. And right now we decided, let's just do a whole bunch of diatom updates to the MarFERReT. So all you have to do really is have your transcriptomes assembled and deposited at NCBI. It needs to have some kind of stable location where you deposit your thing. You need to know your NCBI tax ID of your isolate. So you need to, sequence your 18S. But at that point, you could just add it to the MarFERReT. And and it's included, and it's really not like Mora was saying, it's really not that hard technically to do that. So, and we're doing that right now. We have like 15 isolates of diatoms, and and then there's this Alverson paper that has a whole bunch of transcriptomes. Like, okay, let's do that too. I'm actually doing that with an undergrad right now. So it's not so hard to add. And I think if any of you guys have these transcriptomes on your computers, we could, as soon as you make it available on NCBI or on Zenodo, then it can be just folded into the MarFERReT. So
thank you, Sasha. Yeah, that's a good point. So I circled back to this slide here, just to emphasize that, expanding it really is just kind of at the bare minimum, you're just adding in a couple fields. You have a new internal reference number. The MarFERReT name is just what we, decide the name is, because a lot of the stuff we look at, names change. Here, look at me, and or there's conflicting sources. So this is what we decide on. We're going to call it this. This is the NCBI tax ID, the one, and if we put some effort into it, we would verify this is updated. And then this just points to the FASTA file and tells us whether we're looking at proteins or nucleotides, and that tells us how to press it further. So again, yeah, like Sasha said, it's easy. You just kind of add lines onto it, and if you do some quality control, which is then you decide, okay, I'm including this or I'm not including it. And it's also one way to kind of have a level of customization control too. But as long as in the end you document, this is what's in there, this is what I did, and then drop it on something like Zenodo or Figshare, and then you have a DOI for some version of it, but it won't be as difficult as building it up from scratch.
Mora, can I ask you what your experience with the NCBI taxonomy has been like? Because although it might be simple to add a reference transcriptome, what takes me a long time is going through and making sure that all the classification levels are matched appropriately across the hundreds of references that are now in that library. For example, is the word masts in one of the levels, and should that apply to this new organism that I'm pulling in? Because then you might want to collate at one of those broader levels. So what was that experience like with you and NCBI? And was that a time sink for you as well?
Absolutely. Yeah, NCBI is a mess. It really is a mess, but at the same time, it's a mess because it is, I think, the largest attempt at collating all this information. And it is this big community project. And yeah, it's difficult, where ranks don't exist for an entire lineage when they do for another. So it's taken me some time to say, okay, we're taking class Dinophyce and phyla this, because the ranks that we talk about as oceanographers don't often line up with what's in NCBI. So it's a love-hate, yeah, absolutely. It's not perfect, but it's also very standard. And yeah, I'm not sure if we have a feasible alternative to it. At what it does. There's the PR2 architecture, which I've tried to bridge here too, but yeah, it takes some time. I do think that maybe we have more leverage over what happens in those decisions than we give ourselves credit for. Actually, because it's community supported, we can go on there and make requests to NCBI and say, hey, can we fix this? Can we add these ranks? And I think that's what Sherry had to do, perhaps, Sasha, correct me if I'm wrong, to get her new isolates added into NCBI. So that is kind of the crutch, if we want to say this is this, we need to have the NCBI thing, the handle to point to.
Yeah, I think the point is, yeah, you have to really show your taxonomy of if your strain is really something new, you will have to provide some evidence to NCBI, and that is really that strain. If it's really, really new, you need to make a new name even. Yeah, it may be a little bit more difficult. You need to give probably multiple lines of evidence, about your new taxonomy. But yeah, my experience with NCBI is that they're pretty responsive when you write email, but yeah.
Sarah Smith, did you have your hand up? Did you have a question or comment or?
Yeah, thank you. Yeah, so I'm wondering kind of what Mora, your thoughts and maybe this whole community's thoughts are on including something like an ortho MCL step in these pipelines, or perhaps like on the database and then just adding that as like a tag to whatever comes out at the end, because we've found it to be really useful in kind of eliminating some of the noise, like these spurious sequences that kind of end up being like poorly annotated protein models or something like that. Like they fall out of the clusters, they become like singleton clusters. And then you end up focusing are able to focus your analysis on some of the things that are core and central to all phytoplankton or all diatoms or all promnesiophytes or whatever it is that your question is about. And then maybe the clustering then itself becomes kind of interesting, like, oh, there's a subset of the diatoms that have, this is a small cluster with only 30 open reading frames, but all diatoms have it, and it seems to be kind of uniquely adaptive in this particular environment. So it's kind of a way of like rather than assigning an identity to every sequence, like a bottom-up way of analyzing a data set a little bit. So have you thought about what that functionality might be?
Not in detail. I think that's really interesting. That's a good point. And, kind of putting this out there in the first take, I did it with PFAM, and I've also used CoFAM too. And a lot of this depends on, what is our approach and our question. Yeah, there's so many opportunities to kind of improve this research and take it new levels, which is exciting why this group is getting together to talk about what the different things we can do with the data, with the references, and kind of get it to an even better place.
Yeah, the problem with like ortho MCL clusters is that they're never static. Like as soon as you add something new in, you have to redo the whole ortholog clustering, which computationally might be prohibitive. But yeah, and might be something that's better done like after the fact, rather than sort of nested within like a pipeline like that. But yeah, interesting to hear your thoughts about it.
I think this is the utility of, nothing needs to be the final version, but tying every step that we do to a data citation, a data object identifier. Here's MarFERReT version 1.1. Here's MarFERReT when I mash it together with Marine MarkerDB. And now that's a new DOI, and it's got its own Zenodo page. And then if you do something with Orthos MCL, you could say, hey, here's the FASTA files I'm looking at, the NCBIs, but you have a different data product. Yeah, maybe it ends up as part of the official MarFERReT package, or maybe you put it up there and you put it on Figshare Zenodo and say it's from here. So I think there's all these avenues to say, okay, yeah, this, and also let's do this too. And I think as long as we can kind of keep these little handholds of the DOIs and everything else to kind of string together the content and the methods that we use to get along, then yeah, that's again my hope, and kind of peppering out profuse hyperlinks everywhere in the text is to yeah, get it accessible. So if you do that, yeah, it'd be really interesting to see what comes out of that, and that could be a really cool accentuation on this, or take it a step further.
Okay, that's awesome. We do need to wrap up. Sasha, do you have a quick comment that you want to add, or?
Yeah,
okay?
I mean, I guess we
Okay.
could talk forever, I guess. No, I just want to say, because when Mora was making this reference database, it was really for taxonomy purposes, right? Taxonomic annotations. And functional annotations came more as a quality control of the entries, of the taxonomy. But now that we have this data set, and, we could think about how we could use it also just to dive more into functionality and how there's different phylogenetic groups, what kind of functionality defines them or separates them. So I think maybe that's actually another layer. We don't use MarFERReT for functionality right now to study, to annotate functions. But it could be an interesting future, avenue.
Well,
Yeah.
maybe we need a small working group that's OCB funded to help us address all this stuff. Thank you guys very much for contributing to discussion. Mora, Sarah, thank you so much for sharing your work today. We really appreciate it. I'm going to post a URL to the webinar links and more information for the Meta-Eukomics group, and there'll be more webinars in the coming months, so keep an eye out. Thank you, everyone.
Thanks, everybody.
Thank you. So nice
Thank
to
you,
everyone.
Good to see everyone.
Enjoy the last
Bye,
breath
thank
of
you.
summer.
Good to see you.